everyone, and welcome back to By Holly G. If you are new around here, because this channel is still very new, I'm gonna say hello. I am Holly Gabrielle, hence By Holly G, and this channel is all about biology, so I hope it makes sense. But welcome to the second official biology video on this channel, and as you can tell from the title, we are essentially going to be talking about stem cells and everything to do with stem cells. It is a very complex field, kind of just like anything in biology, but the thing that makes stem cells really complex and confusing is the fact that not only is the biology very intricate, but many of the terms and the words we use in the field of stem cell biology are very poorly defined, and so that means it's really difficult sometimes to navigate this field. However, we are going to have a very fun conversation about stem cells because I honestly love this field. The first thing we need to do in this video is define a stem cell. Obviously, we cannot do anything else without defining a stem cell, and to make it easier, I'm going to give you two key properties that are key to stem cells that will help us define stem cells. They are cells essentially that have the ability to firstly self-renew, which means that they can make more copies of themselves, and secondly, stem cells have the ability to differentiate, and by that I mean that they can become a more specialized cell type. For example, a stem cell is normally, not in all cases, but normally undifferentiated, and then it will differentiate or specialize into a cell with a particular function. So for example, that could be a muscle cell, a neuron, a white blood cell, a red blood cell, you know, those are specialized cell types. They have particular functions. Just to kind of set the scene and to give you guys a bit of a broader context, without stem cells there wouldn't be development, you know, like a human fetus would not grow to an adult without stem cells. We have things called adult stem cells, and they are basically responsible for the repair and maintenance of basically every single body tissue. Stem cells and their dysregulation plays a role in like degenerative diseases, so for example like macular degeneration, age-related macular degeneration to do with your eyes. They have therapeutic applications in like regenerative medicine, which is really cool, and they also have a link to cancer. We have these things called cancer stem cells or stem-like cells. There are other applications we could talk about, but those are just the main ones I wanted to mention at the start. The thing you have to remember with biology is that the deeper you go into a topic, the easier it is for you to lose sight of the bigger picture. So I think it's really important with every single topic to start with the broader context and to be like, okay, what is the importance of this topic? As usual, if you like this video, definitely give it a thumbs up before we dive into the details. And yeah, if you are new and you want to stick around, you can subscribe and turn the bell on so you know when I upload. But yeah, we're now gonna basically jump into the main details of this video. The first main concept then that we're going to tackle is basically this idea of potency. Potency, we're going to define it. It is essentially the ability of a stem cell to become different cell types. So as I said, stem cells, they self-renew, but they also can differentiate. The analogy that we're going to use is the landscape analogy, and it is actually Waddington's landscape. It is a formalized analogy in biology, and so it's this landscape analogy, and it helps us to understand that potency exists on different scales. We have something that's called totipotent, then we have pluripotent, multipotent and unipotent, and different stem cells will have different potencies. So for example, some types of stem cells will be totipotent, others might be multipotent, etc. But we're going to start at the top with totipotent because that stem cell, a stem cell that is totipotent, I want you to think of it as having the most power sort of thing. What I mean by that is a totipotent stem cell is a stem cell that has the ability to become any cell type, and that includes cell types of the embryo, so embryonic tissues, as well as extra embryonic tissue, and that is what I mean by the placenta. The second definition is pluripotent. So pluripotent is a bit more restricted or slightly less powerful than a totipotent cell, we could say, because it can become any cell type but not those of the extra embryonic tissue, i.e. the placenta. Multipotent is quite hard to define. It can just become multiple cell types, but it is more restricted than a pluripotent stem cell. And then finally we have a unipotent stem cell, and a unipotent stem cell will only become a single type of cell, so that's why it's unipotent, uni meaning one. Now to put this on our landscape, Waddington's landscape, so a totipotent stem cell is going to sit right at the top of the landscape because it can roll down the landscape and become any cell type. And when it becomes a specific cell type, it's going to stop falling down the landscape and it hits kind of like a rock or a valley where it can't move any further down the landscape. So because the totipotent stem cell is right at the top, it has the potential to become any cell, as I said. A pluripotent stem cell is going to sit a bit further down that landscape so that it can fall down and become lots of different cell types, but it is just slightly more restricted, if you see what I mean, because it can't fall down as far. And then a multipotent stem cell will sit a bit further down, and then unipotent will be sitting like quite near the bottom because it literally just rolls down into one valley because it can only become one cell type. I hope that makes sense, but that is potency, and that is the Waddington's landscape analogy. At this point, I also think it's important to give you some examples of stem cells of different potencies. Let's again go back to a totipotent stem cell. The only cell that we know for sure is totipotent is the zygote, and the zygote is the single cell that is formed upon fertilization. So when a sperm cell fuses with an egg cell, we form a zygote. That single cell is totipotent. It can become any cell type, as I said, embryonic tissue and extra embryonic tissue. During development, that zygote, that single cell, then starts to divide. It forms the embryo, which is a ball of cells, and it will develop into a fetus and then a full-grown human being. But that zygote and the first few cells that are formed in the first few divisions, we think they are also totipotent. Okay, but the cells that then form after that during development, as the embryo continues to divide, they are then pluripotent, so they lose the ability to become placental tissues. They then will eventually disappear. So during development, pluripotent cells are transient. As a fully grown adult human being, you will not have any pluripotent cell types anymore, because adult stem cells, so we all have adult stem cells in our body and they reside in different tissues, but they are multipotent at max. Adult stem cells are either only multipotent or unipotent. The best known adult stem cells are hematopoietic stem cells, HSCs. They are the stem cells that reside in your bone marrow, and they are responsible for producing all of your red blood cells, white blood cells, and platelets. Hematopoietic stem cells are an example of a multipotent adult stem cell. An example of a unipotent adult stem cell is a muscle stem cell, which has a particular name. They are called satellite cells, and they normally sit on the outside of bulk muscle tissue. Adult stem cells are very, very interesting. As I said, their function is to maintain tissue and repair tissue. We think that they exist in pretty much every single tissue of the body. However, identifying adult stem cells is very, very complex and very difficult to do, because it's not as if there are any key defining characteristics of adult stem cells that we can look for. They are all very different in different contexts, which makes it difficult. Before we move any further, I want to define a term that is going to be used in this video. I am going to use it. It is the idea of a progenitor cell. So we've talked a lot about stem cells. We've talked about different stem cell potencies so far, and the fact that different types of stem cells have different potencies or potentials to become different types of cells. We're now going to talk about progenitor cells. They are also known as transit amplifying cells, but what a progenitor cell is essentially very similar to a stem cell. However, a progenitor cell cannot self-renew. So whilst the stem cell has the ability to self-renew and differentiate, progenitor cells cannot self-renew. They can only differentiate. That's all I'm going to say about progenitor cells now. Just keep in mind that they are not stem cells. They're very similar to stem cells, but they just can't renew, or they have a very limited capacity to self-renew. So keep that in mind, because we're now going to talk about how stem cells divide. So we have three types of divisions that a stem cell can undergo, and when a cell divides, it just basically makes two new copies of itself. The first is a symmetric self-renewal division. So a single stem cell will divide and make two more stem cells. So it's just self-renewing, and it's a symmetric division because the two daughter cells that are made are identical. The second type of division is then a symmetric differentiation division. I don't know if that's the official term, but we're going to say symmetric differentiation division. What essentially happens in this case is the stem cell, instead of producing two identical daughter cells that are also stem cells, it will produce two identical cells that are slightly more differentiated. So it's not self-renewing in this division. What it will be doing is it will be producing two progenitor cells normally, and those progenitor cells, they're slightly more differentiated than the stem cell, and they will then go on to form, through quite a few rounds of division, the final specialized cell type. So as those progenitors divide, they become more and more committed to the cell fate that they are going to become. The third type of division is an asymmetric division. So the stem cell will make another stem cell, and a cell that is more differentiated. Normally, as I said, a progenitor that will go on to differentiate. So that is an asymmetric division because the two daughter cells are different. And the next thing I need to emphasize is that in a population, in a tissue system, you always have to have some stem cells, and you also have to have the cells, the specialized cells of that tissue. So for example, in a muscle, you need to have the muscle stem cells, and then you also need to have the specialized muscle cells that are gonna do the work in the muscle, like, you know, undergo contraction and relaxation. And so what we need to now think about is how can we maintain that balance? How can we always ensure that we have stem cells and differentiated cells? So there are two models to describe this. The first model is called the deterministic or hierarchical model, and this is a very kind of like simple system. Some stem cells, what they will do is they will always undergo an asymmetric division, and very simply, that will always ensure that you have stem cells and more differentiated cells if you're always undergoing that asymmetric division. The second model is a bit more complex, and this is a stochastic model. This model is basically saying that you will have a balance of all of those three types of divisions that I've described. So sometimes the stem cell will divide asymmetrically. Sometimes it will undergo a symmetric self-renewal division. Other times it might undergo a symmetric differentiation division. But essentially there always has to be a balance of the three types of division, because overall then you're going to have stem cells and differentiated cells in your population. The type of division that the stem cell undergoes is not always the same. It's stochastic, kind of like random. There is an element of randomness to that system. That kind of understanding came from studying hematopoietic stem cells, and the hematopoietic stem cells are a very good example of the stochastic system. They don't always divide asymmetrically. That would be the other model. That is the hierarchical model. And basically every stem cell and stem cell system will be different. Sometimes the stochastic model will apply, other times it will be the hierarchical model. There might even be other models that we don't know about. The next thing I'm going to mention, I'm not going to talk about too much, but I just wanted to mention the stem cell niche, just in case, you know, you were interested in this, or in case you want to go away and do a bit more research. You know, stem cells, they don't really function as cells on their own. They need kind of like support, and that is what we call the stem cell niche. What the stem cell niche is doing is essentially it's just helping the stem cells, and it's regulating their differentiation and self-renewal. And the niche can take the form of being, you know, surrounding cells, like actual cells that kind of support the stem cell, and or soluble factors that are secreted and that tell the stem cell what it needs to do. I would just remember it as the kind of environment that the stem cells find themselves in. We're now going to jump back to development, and we're going to talk about embryonic stem cells, which a lot of people have probably heard of. So yeah, this is a really important thing to understand, and it's something that you can get confused with. Yes, they are obviously stem cells, so like any other stem cell, they're going to be able to self-renew. They can divide to produce more differentiated cells, but essentially embryonic stem cells are pluripotent. So remembering our definition of pluripotent, it means that they can become any cell type except cells of the extra embryonic tissue, which is basically the placenta. So we have some technical words coming in, but the embryonic stem cells are basically in vitro artifacts. They are harvested from the embryo. We then culture these cells in the lab, and we keep them as embryonic stem cells. We make them stay in a pluripotent state, because during development, the pluripotent cells of the embryo won't stay pluripotent. As I said at the very start of this video, they are only transient. They will eventually disappear, and in the adult, we will be left with only multipotent stem cells. So going back to our zygote situation, we've had fertilization, we have this single zygote. That is totipotent. It starts to divide to form this ball of cells that we know as the embryo. Some of those cells will be totipotent, but the majority of them are then going to become pluripotent. In embryology and developmental biology, we characterize different stages of the embryo as it develops, and there is one particular stage called the blastocyst, which I'm going to show you a picture of. This whole thing, this ball, is the blastocyst. It has this outer layer of cells that we call the trophoblast. We have this space that's called the blastocele, but don't worry about that. And most importantly, we have the inner cell mass, and that is a region of cells, a cluster of cells that is going to be labeled. They are the pluripotent stem cells, and to make embryonic stem cells, as I said, they are cultured artificially in the lab. So if we take a blastocyst and we take out those inner cell mass cells, we then culture them in the lab, and we keep them artificially in a pluripotent state. So basically we prevent them from fully differentiating. Whereas naturally during development, as I said, those inner cell mass cells, they are going to be responsible for forming the other specialized cell types, but they're eventually going to disappear during development. There is a fixed time point at which there will be no more pluripotent cells left. So 14 days after formation of the zygote, you will no longer have any pluripotent cells left in that embryo. At day 14, you start to see the appearance of your adult stem cells that are multipotent, and they will exist for life. But what I'm saying is those pluripotent inner cell mass cells, they are transient in development. They exist temporarily. However, when we take them out from an embryo, we artificially harvest them, and then we artificially culture them in the lab. What we have to do is we have to feed them like factors and make sure that they are in a certain environment that prevents them from differentiating. So we artificially capture them in a pluripotent state. And that is why we say that embryonic stem cells are an in vitro artifact, because they don't exist naturally. So going back to then a definition of embryonic stem cells, what I would say is that embryonic stem cells are pluripotent cells that are derived from pluripotent inner cell mass cells from the blastocyst stage of embryonic development. That is such a mouthful, but I hope it makes sense. Now you're probably wondering, what's the point? Like, why are we keeping these embryonic stem cells in a self-renewing pluripotent state? Like, what are we gonna do with them? And that's what we're gonna talk about now. So the uses of embryonic stem cells, essentially. We can use them in multiple ways. I'm gonna mention 3 key uses of embryonic stem cells. The first is to make genetically modified mice. We will genetically modify those embryonic stem cells. Genetic modification could be a whole other video. We would then insert those cells into a mouse embryo, and then we would generate a fully grown genetically modified mouse. The second use is basically for regenerative medicine that I mentioned at the start. So we have these embryonic stem cells in the lab, and what we can do is by culturing them in different conditions and giving them different factors, we can make those embryonic stem cells differentiate into basically any cell type that we want. Not the extra embryonic tissue, because they are only pluripotent, but by giving them different factors, we can tell them to differentiate into a particular cell type that we can then insert into the body to cure a degenerative sort of disease or condition. For example, age-related macular degeneration is basically, with age, your eyesight will deteriorate because of degeneration of a region of the eye called the macula. If we basically differentiate embryonic stem cells into cells that can be inserted into someone's eye, in this case specifically, they're differentiating the embryonic stem cells into specific cell types of the eye. So it could be photoreceptors, or it could be the retinal pigmented epithelial cells. It could be either of those cell types, but we would make those specialized cell types and then reinsert them into someone's eye, and theoretically someone's vision could be cured. There are lots of problems with regenerative medicine at the moment. There are problems with cell delivery and cell integration. How do we get those cells to the target organ, and how do we get them to stay there and embed and become part of the normal tissue? Problems associated with the risk of cancer development, because if you have your embryonic stem cells, you've differentiated them supposedly, how do you know that they're fully differentiated? If they are not fully specialized, then they may still retain some stem cell like properties, and that may mean that they have the ability to proliferate and self-renew, and that is basically gonna lead to cancer. So there is a risk of tumor development. There is also a risk of zoonoses. So in the lab, if we're culturing these cells, we use quite a lot of animal products in culture. So there are risks of animal diseases being transferred to humans. That's a zoonosis. A big problem is that when we do these things in vitro, when we differentiate the embryonic stem cell into a specialized cell type, that's all being done in a lab. And that is never ever going to recapitulate what actually happens normally in development. The actual body is like 3D, for example, and there are lots of forces that will influence stem cell differentiation. In the lab, if we're doing it in petri dishes, it's kind of like 2D, and we're not going to be mimicking that actual situation very accurately. So what I'm trying to say is that in vitro, in the lab, we are forcing the differentiation of these cells. It's going to be a lot faster than it normally occurs. Is it going to be done correctly and accurately? Probably not. The final thing is to do with like rejection. So if you've got embryonic stem cells, often the cells that we differentiate from the embryonic stem cells, when we insert them, they are often going to be rejected because they are going to be recognized as foreign by the recipients immune system. We don't have embryonic stem cells for every single individual. We cannot get around this problem because I would need to use my own embryonic stem cells to make the cell types that I need to avoid immune rejection. That's an impossible feat though. That's never going to happen. The third use of embryonic stem cells is then drug screening and disease modeling. So in vitro in the lab, we would basically again differentiate our embryonic stem cells into a particular cell type that we want to study, and then we would, yes, like model that disease, and we would look at how the cells behave. We would also do drug screening by applying different drugs and see if they can correct the cell function sort of thing. So that is the third use basically. So embryonic stem cells basically, they have those three uses, just to summarize. As I said, there's quite a lot of problems with using embryonic stem cells, in particular for regenerative medicine. Embryonic stem cells as well have quite a few controversial issues. So we basically have an alternative. We have now a solution. We have things called induced pluripotent stem cells, iPS cells, and they were discovered, or they were first made, by a person called Shinya Yamanaka. I cannot pronounce things, as I've said before, and will always say time and time again, basically Yamanaka I'm gonna say. These are reprogrammed cells, and it's really, really clever technology, because what we're basically doing is we're taking a fully differentiated cell and we are reprogramming it, or we're turning it back into a pluripotent stem cell. We are reversing differentiation. We're going to go back to our landscape analogy, the Waddington's landscape, because in this case, instead of the ball rolling down the hill or the cell rolling down, we're pushing the cell back up, because we're taking a cell that is fully differentiated and turning it back into a stem cell. We're pushing it back up that landscape. This, as I said, it was discovered by Yamanaka. He first managed to do this using a fibroblast. So a fibroblast is a type of differentiated cell, and he reprogrammed this fibroblast into a pluripotent cell that we now know and call induced pluripotent stem cells. He did this with 4 factors. That's 3, Holly. He did this with 4 factors. So the 4 factors are now called the Yamanaka factors. If you are at all interested, the 4 factors, their names, they are all proteins. They are called SOX2, OCT4, KLF4, and c-MYC. We have a fibroblast. We apply those 4 factors. They are proteins, as I said. They have a specific function, which we're not going to get into, but we apply those 4 factors. We make a pluripotent cell that we know is an iPS cell. And this technology has now advanced. We have lots of different variations, so we can apply different factors. It doesn't have to be those 4 Yamanaka factors, the 4 original ones. And we can also do it with lots of other differentiated cell types. So it doesn't always have to be a fibroblast now, it can be a different type of cell. And we can reprogram it to an iPS cell. We basically have cell types that are pluripotent, and they can be used in the same 3 ways as embryonic stem cells. So they can be used to make genetically modified mice. They can be used in regenerative medicine, and they can be used for disease modeling and drug screening. They are essentially, as I said, a counterpart to embryonic stem cells. In particular, looking at regenerative medicine, as I talked to you about, there are lots of issues with using embryonic stem cells for regenerative medicine. iPS cells solve quite a few issues. First of all, they're not as ethically and socially and politically controversial like embryonic stem cells. Most importantly, however, if you use iPS cells from yourself, so for example, say if I needed cells for my eye to treat macular degeneration, theoretically what we could do with this iPS cell technology is I could take my skin cells, we could turn those back into my iPS cells. We could then differentiate those iPS cells into the photoreceptors or the cells of the eye that I need, inject them, and they wouldn't be rejected by my immune system because they have originally come from my skin cells. So we are going to favour the use of iPS cells nowadays instead of embryonic stem cells. However, iPS cells do still share lots of the other issues that I mentioned. So problems with cell delivery, cell integration, risk of tumour development, zoonoses, and the kind of mimicking of the differentiation process. Again, it is still artificial. However, they have made a huge leap forward. What we can do with iPS cells as well, in the context of not regenerative medicine now, but in disease modelling and drug screening, we can use iPS cells for patient-specific disease modelling and drug screening. The final thing to mention about iPS cells, yes, it all sounds amazing, but there are problems with iPS cells. So iPS cell technology, where we're reprogramming differentiated cells. Essentially, this reprogramming process, it's really, really slow at the moment, and it's also really inefficient. So our goal is to make a fully reprogrammed iPS cell that is completely pluripotent and that has lost all of the characteristics of the specialized cell it came from. However, that process of reprogramming is not often fully complete, and so that has lots of issues associated with it. We're trying to understand why it's not complete, how we can make it complete. So there are lots of things we need to do to solve that issue, but I think we're making steps in the right direction. So yeah, that's stem cells. That is everything I wanted to mention in this video anyway about stem cells. I hope you followed it. I hope it was interesting, and I hope you enjoyed it if you made it to the very end. As I said at the start, definitely like this video if you enjoyed it. Comment down below if you have any questions or if you wanted to say anything at all. This is not just going to be the only video I do about stem cells. Any other future videos will just be added to this playlist, but I wanted to give a big kind of broad overview of stem cells. And yeah, I think that's everything to say. If you want to subscribe, definitely hit the subscribe button. I will speak to you very soon in another video on this channel on more biology from Holly G, and yeah, I will say bye. Bye!